module PowerPoint for module 7.2.1. Getting into our section on electrophysiology and specifically the neuromuscular junction here. Keep in mind that this is just kind of a rehash of what we've learned, just going into a little bit more detail. All of the modules in 7.2 are going to be like that. So you've already seen these topics before, but this is just going to go a little bit more in depth into those topics. So starting off with skeletal muscle contraction here, we've talked about the four basic overall phases of it. We talked about 1, excitation; 2, the excitation contraction coupling cycle; 3, the contraction cycle; and 4, the relaxation cycle. And so an axon may branch into control more than one skeletal muscle fiber, but each muscle fiber only has one neuromuscular junction. And at that neuromuscular junction, the axon terminal of the neuron lies near the motor end plate of the muscle fiber. So we'll kind of cover the events of the neuromuscular junction, even though we've seen that before here. So in the first event here, the cytoplasm of the axon terminal contains vesicles filled with molecules of ACH. And acetylcholine is a neurotransmitter, which is a chemical released by a neuron to change the permeability or other properties of other cell plasma membranes. And synaptic cleft of the motor end plate contains molecules of the enzyme acetylcholinesterase, which breaks down ACH. In the second step, the stimulus for ACH release is the arrival of the electrical impulse or the action potential at the axon terminal. And so an axon potential is a sudden change in the membrane potential that travels along the length of the axon. The third step, when the action potential reaches the neuron's axon terminal, permeability changes its membrane trigger to the excitosis of acetylcholine into the synaptic cleft. So excitosis occurs when the vesicles fuse with the neuron's plasma membrane. In the fourth step, ACH molecules diffuse across the synaptic cleft and bind to ACH receptors on the surface of the motor end plate. ACH binding alters the membrane's permeability to sodium ions. And because the extracellular fluid contains a high concentration of sodium ions and sodium ion concentration inside the cell is low, sodium ions rush into the cytosol. In the fifth step, the sudden inrush of sodium ions results in the generation of that action potential, which we'll talk about a little later, in sarcolemma. ACH is removed from the synaptic cleft in two ways. So ACH either diffuses away from the synapse or it's broken down by acetylcholinesterase into acetic acid and choline. And this removal inactivates the ACH receptor sites and the muscle fibers pictured here indicate the propagation of the action potential on sarcolemma. So this is just talking about some of the components of the neuromuscular junction here. And we talked about specifically what the neuromuscular junction was, which is where a nerve terminal interacts with a muscle fiber at the motor end plate. We also talked about the synaptic terminal, motor end plate, and synaptic cleft. And this just highlights what we just talked about. We've also talked about the importance of ACH to this point, and this just highlights the structure as a little bit more about the involvement of ACH. And we'll talk more about this later on as well. So this is an overview of skeletal muscle excitation, and the link between the generation of an action potential in the sarcolemma and the start of muscle contraction is called that excitation-contraction coupling. And this coupling kind of occurs as a triad, and on reaching a triad, the action potential triggers the release of calcium from the cisternae of the sarcoplasmic reticulum, and the change in permeability of the sarcoplasmic reticulum to calcium is temporary. It lasts only about 0.03 seconds. Yet within a millisecond, the calcium concentration in and around the sarcomere reaches 100 times the resting potential levels. And because the terminal cisternae are located at the zones of the overlap where the thick and thin filaments interact, the effect of calcium ion release on the sarcomere is almost instantaneous. Troponin is the lock that keeps those active sites inaccessible. Remember that. Calcium is the key to that lock, and recall that troponin binds at both actin and tropomyosin, and the tropomyosin molecules cover the active sites and prevent that interaction between the thick filaments and the thin filaments. So each troponin molecule also has a binding site for calcium, and it's this site is empty when the muscle fiber is at rest. And calcium binding changes the shape of that troponin molecule and then weakens the bond between troponin and actin. And troponin molecule then changes position rolling the attached tropomyosin strand away from the active sites with its change, and the contraction cycle begins. And so we can go through the steps of skeletal muscle excitation here that you see listed on the screen. So, just in a shortened detail here, step one, the arrival of the action potential. Step two is the release of ACH. Step three, ACH binding the motor end plate. Step four, the appearance of the action potential. And step five, return to the initial state. So this is just a brief overview of the action potential. I don't want to get too deep into this because module 7.2.4 covers the action potential in detail. Just know that this plays a big role in everything that we're talking about with skeletal muscle interactions here. So I highlighted a little bit about excitation, contraction, and coupling, but just to rehash it, remember the action potential reaches the triad, releases the calcium, and triggering the contraction, and that requires the myosin head to be in the cock position, and it's loaded by ATP. During the process of contraction, that neurostimulation of the sarcolemma happens and causes that excitation contraction coupling that we just talked about. Remember, the cisternae of the sarcoplasmic reticulum releases that calcium, which again triggers the interaction of the thick and the thin filaments, and that uses ATP when it produces tension. This just highlights some of the steps that will initiate the muscle contraction, which we talked about. Remember, ACH is released, the action potential reaches the T tubules, the SR releases calcium, the active site exposes and cross bridge formations occur, and then contraction cycle begins. Conversely, these are the steps that end a muscle contraction. So ACH is broken down, the sarcoplasmic reticulum reabsorbs calcium, the active sites are then recovered and cross bridge formation ends. That causes the contraction to end, and then that causes eventual muscle relaxation. We've kind of talked about exposing the active sites already. Remember, the action potential of the transverse tubule reaches that triad and causes the release of calcium ions from the cisternae to the sarcoplasmic reticulum into the sarcoplasm and around the zones of overlap of the sarcomere. Remember, calcium will then bind to troponin on the thin filaments, and troponin will pull the tropomyosin off of the active site so that the cross bridge can form. We already talked about some of the 5 steps of the contraction cycle, but just to rehash them here. Some of the exposure of the active sites, the formation of cross bridges, pivoting of the myosin heads, detachment of cross bridge cycling, and the reactivation of myosin. And this just overviews the contraction cycle itself. In step 1, the actin, which is not bound to tropomyosin at this time, binds to myosin by its active site. In step 2, the cross bridges are formed. In step 3, the myosin heads have been pre-primed for the movement of ATP. And then upon binding of actin, the myosin head pivots, and which will eventually result in what we call a power stroke. In step 4, the myosin ATPases use ATP to break down the cross bridges, which releases the myosin head from the actin active site and resets the myosin head pointed away from the M line. In step 5, the myosin head is now primed to interact with a new active site on the actin, and myosin carry out the 5 power strokes per second while calcium and ATP are available, and each power stroke shortens the sarcomere by about 1%. Let's just talk about fiber shortening a little bit. Remember, the contraction duration kind of depends on one, the duration of the neutral stimulus, the number of free calcium ions in the sarcoplasm, and the availability of energy. And then overall, we'll get to relaxation at the end. Remember, calcium is reabsorbed by the sarcoplasmic reticulum. Calcium ions then detach from troponin. Troponin without calcium pivots towards tropomyosin and back into the active sites on the actin so that no cross bridges can form and covers that active site. Sarcomeres will stretch back out due to 3 reasons: gravity, opposing muscle contractions, and the elastic recoil of the titin proteins that are inside those sarcomeres. And the result is the muscle returns to its resting length. This is just an overview, just a little review. Right, again, some of the disorders that are involved with this. So you can have rigor mortis, which is a fixed muscle contraction after death, and it's caused when the sarcoplasmic reticulum cannot reabsorb calcium, either because the ion pumps cease to function and calcium builds up in that sarcoplasm. When that happens, tropomyosin kind of frees the actin in this situation, and the cross bridges prevents them from forming. And then no ATP can detach the myosin head because ATP is already used up and not available. So those don't detach, and so the muscle never actually relaxes. Here are some more disorders that we can talk about here. You can have botulism or Botox, and that's just the bacterium Clostridium botulinum. And that's a toxin that prevents the release of ACH at the neuromuscular junction. You can have tetanus, and that's the stimulation of motor neurons. And then myasthenia gravis, which is an autoimmune disease, and that causes the loss of ACH receptors, so the muscles become non-responsive to ACH that's released into the sarcoplasm.